Okay. So, summary, eukaryotic mRNA processing. We have a gene, we get a primary transcript of the gene. A 5' cap is added. In this case, for the ovalbumin gene, 7 introns are excised or spliced. Splicing, cleavage, and polyadenylation occur. So we have our extra RNA, our poly A tail. And after splicing, cleavage, and polyadenylation, we have our mature RNA transcript with our 5' cap, our exons joined, and our poly A tail. The likely effect of a mutation in the sequence of AAU AAA in eukaryotic mRNA. So the AAU AAA is required for recruitment of the polyadenylate polymerase. So if we are lacking the consensus sequence that recruits the polyadenylate polymerase, then we are not going to have a poly A tail. And this sequence is also needed for the endonuclease that cuts the end, then the polyadenylate polymerase is recruited and the poly A tail is added. So then the RNA, which lacks the poly A tail, may not be able to leave the nucleus because the poly A tail is part of the mRNA leaving the nucleus. And if the RNA can't leave the nucleus, then translation might not occur because it doesn't leave the nucleus. And we know that translation only occurs outside of the nucleus in the endoplasmic reticulum. It's important to note that there is differential mRNA processing. So there may be multiple sites of poly A where we can have a poly A tail. So there are variable domains, especially in IgG heavy chains, where there are 2 points where you can have poly A sites, and there are special proteins that help direct the choice of poly A tail. And there wasn't a lot of detail about that in the book, but it just said certain enzymes may promote poly A site 1 and certain enzymes may promote poly A site 2. So if there are multiple sites where you can have polyadenylation, then you can increase variability in the genome and in the antibodies that are created, and have a greater defense mechanism in terms of antibodies. And in eukaryotes, diversity in the genome is healthy. So cleavage. So we're showing we have primary transcript and cleavage and adenylation at A1 versus cleavage and adenylation at A2, and what the mature RNAs look like in that case. Another example of where we can have differential mRNA processing. The myosin heavy chain in Drosophila can have alternative splicing, not alternative poly A, but alternative intron splicing. So an intron could be shorter or longer, or there could be multiple introns that are removed and multiple introns that are left in the transcript. So for example, here we have the splice sites for the introns and the poly A site. So a primary transcript has our splicing sites. Splice site 5', 3' splice sites So we could take from this 5' to the first 3' as our intron, or you can take the 5' to the second 3' as the intron. And you get 2 transcripts based on that. So we have removal of the short intron, the 5' to the early 3' site. And then you have removal of the full intron, the 5' of the intron all the way to the second intron stop. So we get differentially sized mature RNAs based on how much of the intron is removed. And this differential splicing serves to increase genetic diversity. So we also have differentials exon sizes when you notice that, because if we only take the first portion of the intron and we leave this second portion that is potentially an intron, then we have a differentially sized exon at the downstream. The downstream exon is larger in that case than it is when you remove the 5' to the second intron stop. So alternative exon splicing also exists. So we have a second exon here or a larger exon, so smaller exon, larger exons, differentially mutually exclusive alternative exons, and etc. that lead to different sizes in the primary transcript, as well as having alternative sites for the poly A tails. It ultimately leads to a large amount of genetic diversity. Okay, differential RNA processing. The pathway is chosen by processing factors or RNA binding and RNA binding proteins. So the processing pathway, which sites are spliced, where we polyadenylate, this is chosen by our processing factors and RNA binding proteins. So whether you remove intron 2 or whether you remove the intron between 1 and 2 or etc. is going to differentiate what your mature RNA looks like. This is an example of the, what's this gene called? The calcitonin gene. The calcitonin gene codes for 2 different proteins. It codes for calcitonin, and it also codes for calcitonin associated protein. So which is called CGRP. And this is necessary for thyroid. One is used in the thyroid and one is used in the brain. And the calcitonin protein has differential splicing than the CGRP. For the calcitonin, the last intron, it is polyadenylated after 4. We have exon 1, exon 2, exon 3, and exon 4 in blue. And the calcitonin gene stops and is adenylated after exon 4. And the splicing sites are the intron between 1 and 2, the intron between 2 and 3, and the intron between 3 and 4. And that leads to the mature RNA and the following protease action, the calcitonin protein. Cleavage at the second polyadenylase site includes all 4 introns and the late polyadenylation. So the CGRP is a longer transcript, and splicing cuts out the intron between, includes 4 as part of the intron between 3 and 5. So this entire area from 3 to 5 is considered an intron when the CGRP primary transcript is processed, so that the exons that lead to the CGRP protein are 1, 2, 3, 5, and 6, and then translation in protease action results in our CGRP protein.